Good evening, everyone. It's my pleasure to introduce our silver medal winner and tonight's speaker, Professor Knox Makunga. Since joining Stellenbosch University in 2005, Professor Makunga pioneered medicinal plant biotechnology research in her department, building a world-class laboratory and research program. She's mentored over 53 graduates at the PhD, MSc, and honors level, many of them graduating cum laude, and continues to attract students from across Africa and internationally. Knox's research program her research impact is substantial, She's had over 75 peer-reviewed publications, holds 6 international patents, and is a founding member of Fienti Products, which translates botanical research into commercial natural products. She collaborates with researchers globally and has contributed to the UN Environmental Programme's publication on measuring progress towards SDGs. Beyond research, Knox is a champion for making botany accessible to everyone. She founded LeafApps Initiative, which brings learners from underserved schools in for botanical training, and also co-founded the global movement Black Botanist Week, which amplifies voices of Black, Indigenous, and people of color in plant sciences. So please welcome me in joining Prof. Knox Makunga. Thanks so much for joining us, Knox.
Thank you so much. I hope everybody can see my screen and hear my voice. So I'm going to get straight into it. Today I'm pretty much going to talk about South African medicinal plants and biotechnology, and I'm going to take you through a journey that will outline some projects that we currently have in our lab and some past projects, and then I'll also give some ideas about what we want to do in the future. Okay, so hopefully the audience that's joined realizes what a wonderful heritage that we have here in South Africa and how incredible our plants really are, because we have these biodiversity hotspots that are chock-a-block full of plant species, which happen to be unique. And we are amongst the world's centers of mega biodiversity. And I sit in the Cape Floristic region with about 9,000 species in this really small area. So it's really a concentration of flora in this part of the world. And this, of course, is pretty close to the succulent Karoo, with 10,000 succulent species and many different geophytes. And very recently, I actually went up to Nieuwoudtville to go and check out these bulbs as they were actually flowering. And I originally come from this part of the world, the Maputoland, Pondoland, Albany corridor, and that's known to have about 1,900 plant endemics. And so South Africa and Africa as a whole is world-renowned for its flora. And some of those plants are extinct in the wild, such as the wild ginger that I've indicated here. And so this pretty much threatens the conservation of this particular species. And in one way in which this plant has been saved is through the application of tissue culture. That is a second generation biotechnology. I'll highlight here Erythrina caffra. I've seen them flowering here in Stellenbosch at the moment, and I'm sure in places like KwaZulu-Natal, they have been going probably since late July, but they have these very characteristic, beautiful, coral-colored flowers. And recently this name has had a twist in its name change, and now it's known as Erythrina afra. And the velvicias, of course, are found in the semi-arid areas of South Africa, including Namibia. They are really, really quite charismatic. 2 leaves that sprawl out, takes them a long time to grow. And last but not least, I just want to highlight Aleodendron dichotomum, which marks areas of the Namaqualand with its splendor. And so our plants are unique in their character. They're pretty beautiful. They're found in these geographically interesting areas. And just to delve a little bit into this flora, I'm going to show you some of these wonderful plants that are home here in South Africa. And many of them are found in very unique areas. They're very unique in their character. They've fascinated historians for generations. They are also globally found as horticultural species throughout the world. And geographically, the climates that they're actually found in are very interesting, and they have evolved mechanisms in order to be able to live in those particular environments. And some of those climates are pretty harsh. Here in the Cape, you have areas that have got low nutrition, where nitrogen and phosphate is really, really scarce because the soils are ancient. And all of this, including different pollination syndromes, has led to this explosion of plants that we see in South Africa. And of course, people have been interacting with these plants for generations. So some of the oldest explorers of South African flora and perhaps South African ethnobotany, this very good looking gentleman here, Pape, he was amongst the first people to record information about South African plants in relation to its ethnobotany during the days of the Cape Dutch East India Company. And some of these plants landed in centuries past in places like Holland, where now they are found in different parts of the world as horticultural species. So from a medicinal perspective, there has been a recordal of histories linked to medicinal plants, and this is actually taken out of one of Pape's documents, where he indicates that the local people, the indigenous people at the time, were quite agreeable in tending to the sick in the ships during the explorations of the Dutch East India Company. And so our plants are globally renowned since the 1600s, but that information that is linked to these plants has been held by local people for centuries, and it still carries importance even till date. And nowadays we have a good appreciation that an area such as South Africa with a large plant biodiversity somewhat also holds a lot of biochemical diversity. And when you include the cultural and religious and psychospiritual aspects that are attached to human ecology, this actually leads us to opportunities in economic botany. And down here in the Cape, you've got two major groupings of peoples that are important in utilizing the flora that's around them and even bringing in species from other areas, such as the possums, who actually sometimes take plants from the Maputo-Pondoland Albany corridor and actually bring them into the city centers. And sometimes they also interact with the Cape bush doctors or the Bosse doctors, who are the main commodifiers of traditional medicine here in the Cape. And this ancient cultural practice has been brought into contemporary society, and it is dynamic and it's forever changing. And this actually leads us to some of the reasons that make it so unique. Okay, I think my cursor is doing things here. So it's quite important to better understand why it's important to study medicinal plants in a modern context. And I think this particular YouTube video or documentary is a very interesting one. I'm not necessarily going to play it tonight because there's quite a lot of information that I'd like to go through. But if you'd like to listen to this and see this and, learn some lessons from this, it pretty much outlines why it's important for South African healers to still practice this ancient culture of healing in a modern context. And you've got some testimonies there from various different people who give accounts of why they still interact with medicinal plants in this modern time. It's very interesting, so I do suggest that you go and listen to it at some stage. So in South Africa, I think most of us are quite well aware that the medicinal plant trade is really split up into a informal trade as well as a formalized trade, and that the healthcare system is also very much reliant on medicinal plants. At the same time, the bioeconomy that is growing in South Africa also relies on medicinal products, such as this particular product, Cold Care. That's how Lintagon is labeled in other parts of the world. And of course, rooibos is very important because it services the livelihood of many different communities who grow rooibos, who harvest rooibos, who still exploit rooibos as a primary healthcare medicine. And if we try and look at what's happening in the botanical extracts markets, we can see that this is a growing market globally that's projected to reach about 11 billion US dollars in 2030. And I think it's important for us as South Africans who come from this heritage to participate in these international markets by providing exciting new botanical extracts that can actually ultimately also participate commercially in these international markets. Apart from that, if we look at the decadal plan of the DSI, you can see that central to this decadal plan are African natural medicine interventions, and some of the areas that need to be further developed include medicine bioprocessing hubs, good clinical trial practices, high-end manufacturing practices, as well as meeting regulations that are actually stipulated by SAPRA. And this, of course, takes science in order to be able to fit into those different solutions. And one solution which has been part of my career is the exploitation of biotechnology. And I do like this cartoon, and I've shown this a few times at a few conferences. And I'm just going to leave us with the take-home messages here. We can read what this cartoon is saying whilst I speak. And pretty much, the Gogo's asking this young man about wild ginger, and she's concerned that wild ginger is ultimately not going to be available in the future. And this very intelligent young man says, "Don't worry, Gogo, biotechnology can help." And Gogo has no idea what that is, and essentially the young man explains that it is using living things to do tasks and make useful products. And that has been some of the ideas that have formed the basis of my particular research. So if we now look at plant biotechnology and we focus on global patterns as well as South African patterns and the adoption of these different biotechnologies, we reported this in the South African Journal of Botany in the special issue that was celebrating South African botany. So this paper has come out, and do go and find it. I think it's quite an exciting paper. In that paper, we look at different biotechnologies and we quantify papers that have biotechnology as a theme. And you can see that South Africa does have a bubble. So the size of the bubble gives you an idea of how many publications are available. South Africa has a bubble, and it's quite a large bubble in comparison to other Southern African countries, but it's not nearly as big as in other parts of the world. And the largest bubbles are actually coming from the north, where they don't necessarily have that biodiversity. So what kind of work is being conducted globally? In terms of the global biotechnological space, you can see that the topics that are of importance now are linked to using biotechnology in crops, using CRISPR, proteomics, gene silencing, again, very much linked to CRISPR technology, transgenesis, the genetic modification of plants. So those are key biotechnologies that are being exploited in relation to the global biotechnology space. So major topics include bioinformatics, genetic engineering, molecular marker technology, mutation, breeding, and tissue culture. And it is growing in terms of the papers that actually come out. What does South Africa look like? So in the last 50 years, South Africa has been involved in biotechnological interventions, and our topics are different from what is concerning global biotechnology. So climate change has become quite a critical area, biofertilizers, biocontrol, nutrition. And when we start to dissect this, we'll see that some of the plants that are popping up are crop plants such as maize, sea maize, that particular species that are being investigated quite intensely. You do find some other plants such as moringa, cassava, and these are plants that are critical for nutrition, but I'm not necessarily seeing what medicinal plants are sitting in this particular network. And if we look at those 5 different topics, we see again a trend of increasing biotechnological applications, but the bars are different. What's been quite consistent is the application of plant tissue culture, and more recently some studies in genetic engineering, and you can see the influence of bioinformatics in terms of exploration of biotechnology. If you want to know which topics are being studied, some of the most important medicinal plants in South Africa are actually being studied. These will include Sutherlandia, these Sciletium. I'm just pulling up some of those topics that are important, that are being studied, mainly exploring metabolomics, and I'll talk about some of those studies that come from our particular environment. So indigenous knowledge systems are very much part of the bioeconomy strategy. It's important for us to consider what the decadal plan looks like, and biotechnology is one of those avenues that's actually important for research. And more recently, a paper that I was actually involved with outlined the constraints in relation to biotechnology and the de novo domestication of plants and even South African plants, where applications of gene silencing, gene editing are tools that are not necessarily available in terms of the South African environment, because we don't have the basic information, or we don't necessarily have all of the basic information in relation to these plants. So there are other challenges that we need to put in balance when we consider the application of biotechnology. And some of these challenges are linked to the fact that medicinal plants make natural products, and those natural products are occurring at very low levels inside the plant itself, and that they can be highly variable. Season plays an impact. There can often be a geographic restriction where you've got one particular species that's of medicinal importance, but it sits somewhere along a mountain slope that's very difficult to access. And then also, is it possible to make products that are quality assured? Are we having the right codes of practice and conduct in order to move a species from being utilized as part of the indigenous knowledge system to a point of commercialization where we have to develop agronomic practices, post-harvest handling practices that actually challenge the markets in order to provide quality assured goods. So come with me now to unlock this ethnobotanical treasure trove. And I am going to talk about fynbos medicine, but I'll also include plants from other areas as well. I want you to walk along the Roy Plain with me and into my office and meet some of the members of my group. And I will talk more about the work that we actually do here at Stellenbosch University. We utilize an integrated approach, which takes us out into the field. I'm very much interested in the impacts of stress on plants, and that also leads to better understanding climatic changes and their possible alterations to medicinal flora. We use plant cell culture methods. We develop protocols for different tissue cultures as a strategy. And then we also take some of these plants that we've collected from the wild and we pop them into the tissue culture environment. Sometimes I call this the torture chamber because we can manipulate different stresses inside this very controlled microclimate. And then we exploit various omics, metabolomics, genomics, and more recently proteomics as a means to better understand what the plants are actually doing, but also as part of the drug discovery pipeline. So I'm going to talk about various different projects that have utilized this integrated biotechnological toolkit. The first species that I bring along is Salvia aurea, and I bring this because to the fore because this is one of the very first plants that I started to really investigate when I got to Stellenbosch University. We generated tissue cultures of this particular species, and then we started to explore different populations of this particular plant, and we could pretty much resolve different chemotypic differences that were associated with these different populations. And that work was done by Dr. Mpumelelo Nkomo, who is now no longer a plant scientist, but he is an IP lawyer. So the next species that I'm talking about is Dodonea viscosa. This is a species that's actually found in South Africa and other parts of Africa and pretty much throughout the whole entire globe. We think that its center of endemism is the Australia's region, and somehow it ended up in South Africa, and it is exploited by various people for a variety of different reasons. Dodonea is often utilized for its anti-inflammatory effects. It is very much used in influenza treatments. And in our particular case, we looked at this particular plant and we did an anti-cancer assay, and we were able to show that it does have apoptotic activity, so it does kill cancer cells. It also has anti-angiogenic activity. In other words, it reduces the setting of new blood vessels, so that might mean that it might restrict the cancer to a particular area rather than allowing it to migrate to other areas. We looked at both methanolic and aqueous extracts, and this afternoon I was actually listening to a study by a PhD student that was defending her study, and she has been exploring this as an possible adjuvant treatment in combination with doxorubicin for treating various cancers. So we've been working on this and trying to somewhat develop an anti-cancer treatment along with the Cape Bush Doctors organization as our partners. Ms. Hoffman is finishing up her PhD, and she started off by looking at several different populations of Dodonea in the mountains here around Stellenbosch and in coastal areas. And what we realized is that if you put this plant in cultivation and you also collect plants in the wild, the patterns, the phytochemical patterns, are pretty much the same. So we hope that we will ultimately be able to provide a cultivation method that can provide us with quality assured material so we can make a phytopharmaceutic in order to be able to fit into the cancer treatment space. These plants are exceedingly variable. They are exceedingly variable because if you collect plants from different parts of the Western Cape, the Eastern Cape, and the Northern Cape, those plants actually cluster together in what we call the Cape cluster, and the KwaZulu-Natal plants are actually quite different from the Cape cluster, whereas the Mpumalanga plants have some shared similarities between these different clusters of the KZN as well as the Cape cluster. The compounds that are there happen to be flavonoids, so it's very rich in flavonoids, but then we also have a range of terpenes that are very important for the pharmacological activity of this particular species. And there are still many unknown metabolites that we have not necessarily been able to characterize that are found in the Dodonea extract. What's interesting is that the plants also have morphological differences, and they are very much different in terms of their leaf areas. Some plants are very, very thin in terms of the leaves, whereas some of the plants are actually quite broad. And what's interesting is that the plants that come from the Cape cluster have a shared chemistry and are exceedingly different from those plants that come from KwaZulu-Natal. And that just emphasizes on the chemotypic differences that are available here. We have been trying to understand this chemotypic difference from a genetic population structure, and there are genetic differences that seem to be important in delineating these different populations. So now I want to talk about Sciletium tutosum. This is one of my most beloved medicinal plants of South Africa. Sciletium or Mesobranthium tutosum is fast becoming important not only in local markets but in international markets as well. And in this particular case, we took various plants of Sciletium and we placed them in tissue culture, and we explored different concentrations of salt and then did a proteome analysis where we looked at signatures that were being differentially expressed in relation to the proteins. And if you look at this batch of enzymes, plant products, you will see that many of these are linked to central metabolism. There are also some changes that are actually happening there in relation to vascular sorting. So some structural changes are actually occurring there, but a lot of these proteins are affecting central metabolism. what's happening to specialized metabolism, and particularly to the mesembranes. There are variable changes that are happening to the mesembranes, which are the most important group of medicinal compounds in Dodonaea. And some of those salt treatments actually lead to lower levels of mesembranes. What's interesting is that if you lower nitrogen and then but then at the same time you add high levels of salt, this doesn't necessarily decrease the levels, but if you leave the phosphate, you end up with much lower levels. And this information is important for farmers who are trying to get to best practice in terms of the agronomic requirements that this particular species actually needs. What's also very interesting is that there's a lot of variation along the shoots and different leaves of Dodonaea, of Sciletium, actually accumulate different metabolites. And if we look at mesembrane, it is increasing in the stems, and this somewhat alludes to a coordination of the trafficking of mesembrane. Perhaps it's made in the leaves and then transported to the roots, or it's made in the roots and then transported to the leaves. At this particular stage, we don't necessarily know. What's happening in practice is that people are harvesting the leaf material, whereas if you compare that to the way in which it's used traditionally, people pull up actually the whole entire plant and exploit all of the chemistry of the plant rather than going for the above ground parts. It's quite a complex network of metabolism that is under both seasonal and developmental timing. And very recently, a group in Harvard has actually sorted out the biosynthesis of these mesembrane alkaloids, and hopefully there'll be a reference genome that's actually available for us so that we can answer some of these question marks, questions that we have in relation to which way does that metabolism actually flow. What we do understand is the influence of low to high light intensities, as it does have impacts on the chemistry of this alkaloid group. And in some instances, you require higher levels of light to increase some of these mesembranes, such as dimethyl mesembranol, as well as mesembranol, Sciletium F41, which are important in this chemistry that provides anti-anxiety effects. So another study that I want to share with you is one that we've recently done with Kalen Reddy. He did his PhD with me and is now sitting in Belgium as a postdoc. And every time Kalen and I went out to go and find Sciletium, it was quite difficult to find these populations. We jumped for joy and we developed what we called the Sciletium jump. And ultimately he did this jump when he graduated and switched the stick for his certificate. So we visited various areas throughout the succulent Karoo, and we explored different species of Sciletium in these different areas. We also looked at different populations of Tortosa, and ultimately he studied about 181 extracts, and we used a metabolomics-based approach. We had lots of fun collecting. We went to different landscapes. We met a whole bunch of very interesting people who were very knowledgeable, and that was a lot of fun interacting with local people. And Tina Panas was very kind to take us to her family, and from around her area, she had Sciletium and she has been using it for a very long time. And I'm just going to play this video. I hope that I can get it to play. So I'm switching off my okay, that's not behaving very well. Anyways, I'm not going to worry about it now. Just to say that when we were with Tina, one of the things that she actually told us was that she gives it to babies that have got colic, and she squeezes a little bit of the juice directly from the plant, and then she places it in milk, and then she gives it to the baby, and that allows the baby to have a better sleep. And so it was really interesting and very fascinating to talk to Tina, and we learned a lot from her. And I'm going to move past that video because I've already told that story and I don't want to run out of time. So with Tina, it was really fun, and we also met all kinds of other people who took us along different places throughout that region. And what we did with the plants was to then do the metabolomics, and then we started to look at what happens if we explore different elevations that the plants were actually growing under. And you could actually split the plant metabolomics into two different groupings, where you had one group that was at high elevation and another group that was at low elevation. And we then wanted to better understand what is actually happening in terms of the biochemistry. So we constructed other metabolomics visualizations, such as the heat maps that I'm actually showing you here, where some chemotypes had a very wide range of all these different mesembrines, such as the one that we found in Annesburg, De Rist, where these two chemotypes have got more of the minor constituents or minor mesembrine alkaloids, as well as the chemotype from Dricalen. And this was interesting because these minor alkaloids had been previously shown in a study that we had done to bind much better to receptors that were linked to depression, anxiety, and even Parkinson's disease. So we were interested in finding chemotypes that had higher concentration of the minor alkaloids, such as urbetamine, for example. And so these were the chemotypes that we looked at in consideration. And when we again looked at the low elevation and high elevation aspect, we could actually see that many of those chemotypes that had a lot of unknown nitrogen-containing compounds and the groups from the demethylated alkaloid groups were found at low elevation, whereas the ones that have high levels of mesembrine, these are markers that are considered by industry that occur at high levels in these plants, were found at high elevation. So what does this mean from a biosynthesis perspective? Mesembrine is actually produced from tyrosine and phenylalanine as amino acids. These are precursor amino acids that are derived from the shikimic acid pathway. Both of these amino acids are required in order to manufacture the mesembrine alkaloids. When we look at the plants that are actually sitting at low elevation, these plants are able to make dimethylmesembrinone and ultimately also manufacture mesembrine. But they have a higher concentration of this group of mesembrine alkaloids. Whereas those plants that sit at high elevation are exposed to different light conditions, including higher levels of ultraviolet light and high light intensities. And that high light leads to the production of betalains, which are pigments that function similarly to anthocyanins. They are sunscreens. And betalains also occur in beetroot and are exclusively manufactured in the order Caryophyllales. And so these high level plants shuttle tyrosine, they feed it into betalain synthesis in order to make a variety of betalains, which are yellow and orange colored, as well as red colored betalains. And this is due to the stress that they are actually exposed to. But at the same time, they also have the ability to manufacture mesembrinone and mesembrine. So what this is telling us is that there's no necessarily competition for these primary amino acids that are actually important in the manufacture of mesembrine. So that exploration provides us with a much richer understanding of the biosynthesis pathways and systems that actually control the accumulation of metabolites in the leaves of Sciletium. Because as the leaves actually start to senesce, they actually accumulate different mesembrines, particularly mesembrine, and at the same time as they senesce, they also often start to become pigmented and showcase more of those betalains. So that group of studies actually led us to manufacturing clock out as well as a product called Step In. And clock out allows you to have a better sleep, and it is a product that has been manufactured with the idea that you need that cocktail of the minor mesembrine alkaloids together with the main alkaloids. And we are doing this in partnership with the SAN Council, and that is governed by a benefit sharing agreement. So the next species that I want to talk about is cancer bush, or Sutherlandia, and the work that we've been doing in this particular species in relation to biotechnology. So in the past, we again went out to various different parts of the Western Cape and the Northern Cape and even the Eastern Cape, and we conducted a study where we realized that the plants from Zastron and Colesberg had very good anti-cancer activity. This was done by Samkela Zonyane as part of her PhD. And plants from Huntsby and Pelly Beach, on the other hand, have got high amounts of phenolics and flavonoids, but are not necessarily as effective against a colon cancer cell line. So this led us to thinking perhaps it might be differences in the nitrogen, the phosphates that are existing in these different areas that are driving changes in metabolism. and we placed some plants in tissue culture. We started to explore what happens at lower phosphate levels, what happens at high nitrogen levels, as well as what happens at low phosphate levels, and what happens at low nitrogen levels. And it's quite complex because some of these treatments, especially when you take away phosphate, you take away nitrogen, they cluster quite differently and apart from the controls, as well as plants that have been exposed to high levels of mineral nutrition. And there are a lot of central metabolites that are actually changing. Fructose, galactose, all these sugars are actually changing. And we think that the change is because the plants need to be able to adjust not only to the mineral conditions, but they also are osmotically adjusting to stress. And the sugars thus facilitate that particular process. And if we look at this from a proteomic perspective, there are differences in the proteins that are being expressed in plants that are exposed to high levels of salt. For example, 17 differentially expressed proteins that are were actually in fact expressed new, de novo expression. And then some of these are shared, and in the nutrient treatment, about 8 proteins or so are actually de novo synthesized. So there are many different proteins that are actually changing in relation to the expression when you look at the changes linked to nutrient as well as salt treatments. And it's a very complex matrix of a wide variety of different proteins. And if we try and summarize this, we can then start to see that sugar metabolism is changing, nitrogen assimilation pathways are also changing. There are changes to photosynthesis, glycolysis, nitrogen assimilation, the TCA enzymes that are part of that pathway are changing. Heat shock proteins, which is really part of the stress response system in plants, as well as many cell structural proteins actually change. And ultimately, those changes are also expressed at the specialized metabolome level, where sothlandiosides and soia-saponins, which are thought to be important for the anti-cancer effect of Sutherlandia, actually change. So we've also taken other biotechnological strategies where we've actually overexpressed signaling proteins and transcription factors that may change specialized metabolism, or secondary metabolism, where we actually develop tools to generate a hairy root system, and then we study those changes from a morphological perspective. And those signaling peptides, such as taxamin, have changes as well in the biosynthesis of the soia-saponins. So we can create different clones that have variable chemistry, which we could potentially then place in a large bioreactor and provide a value-added product as an extract that could potentially go into industry. And the last study that I want to share with you this evening is on rooibos, everybody's favorite tea. And here, Catherine Wilkinson, she graduated in March this year, looked at wild populations of rooibos as well as a commercial rooibos, and we did some metabolomic analyses. And together with Jolene Brooks, we also looked at the genetic population structure of this set of populations that were occurring in the Northern Cape and the Western Cape. And in that genetic study, we found haplotype differences that could actually split up these plants based on their reseeding or resprouting life history. What was very interesting about this study is that not only were there metabolomic or chemical signatures that could delineate the populations, but there were also genetic differences that were very much site-specific. And some of these populations were very localized. There were genetic blocks, so a little bit of inbreeding. And this is a problem if you're thinking about conserving those wild populations of rooibos for future generations. So rooibos is very complex, and wild rooibos is also exceedingly complex. What was fascinating for me was that there were so many unknown compounds that had never been characterized before that still are unknown in a species that we have been studying for a very long time. So it would be very interesting at some stage to better understand what these metabolites actually are. We took the study a bit further and we looked at a skin cancer cell line and did an anti-melanoma activity assay. We chose 4 different wild rooibos ecotypes, and we compared this to a commercial ecotype. And it was quite interesting to note that pretty much all of the different extracts did show anti-skin cancer activity. We were interested in better understanding how this is actually functioning in the skin cancer cell line. How does the extract actually work? What are the modes of action? So we used a LCMS peptide proteomic approach where we looked at the skin cancer cell line as well as a normal skin line. And we found a massive repository of proteins that were being affected by the rooibos extracts, particularly in that skin cancer cell line. If you look at these data, you will see that the rooibos and the controls actually have similar proteomic patterns. This is a good thing because that means that the rooibos extract is not necessarily disrupting the normal function of a regular skin cell line. However, in the tumorigenic line, the commercial rooibos extract as well as the wild rooibos extract cluster together and elicit changes that are likely involved in the anti-skin cancer effect. And those changes are many. And they form very complicated networks that are leading to changes in mitochondrial function, that are leading to changes in the way in which signaling actually is taking place in the skin cancer cell line. And we can summarize all of these massive changes that are taking place by putting it into this very simplified diagram where we see that GAPDH, which is important for apoptosis, killing of the skin cancer, leads to P53 markers that are also anti-cancer signaling mechanisms that ultimately lead to cell death. At the same time, there is a breakdown in mitochondrial function, and that's important because a tumor cell requires energy in order to be able to keep growing. And so This lowers mitochondrial function, and that will also ultimately lead to lowered cell cycle progression. So all of these different mechanisms, which are internal, are actually elicited by application of the rooibos extract. And we have an idea in terms of how rooibos has got anti-cancer effects. With that, I just want to finish off by saying that this repository of work actually allows us to be able to lead towards new phytopharmaceutic products with a better understanding of what is actually happening as a result of the extracts, and very much targeting very specific chemotypes that we can understand from a molecular biological perspective in relation to how they actually function. And one of the ways in which we hope to do this is then to ultimately lead a product such as Dodonea in collaboration with the Cape Bush doctors. Once we've sorted out how does it actually work, how can we actually grow it, and hopefully we will have a new product in the market. This work requires for a balance in terms of balancing sociocultural aspects, taking that into consideration, balancing that together with the technologies that we have available, and using those technologies to mine new information that's linked to South African medicinal plants. Many of our plants are still not fully characterized, and biotechnological applications have the potential to improve our knowledge in relation to these particular species. And it's exciting that we have new reference genomes that are available, because then this will allow us to be able to better understand how the plants actually function intrinsically. And with that, I'd like to thank you for your time and all the people that have been working in collaboration with us. And I'm happy to take some questions if questions are available.
Thanks so much, Knox, for that presentation. Are there any questions in the audience? You're welcome to raise your hand and unmute yourself or type questions into the chat.
I'm going to stop sharing unless somebody wants to see something again.
I have a question about the rooibos. Oh, go ahead. Sorry.
Sorry, I don't know where to raise my hand here. Thank you, Knox, for that beautiful talk. I just want to check. Yes, in terms of the collection of Dodonea in KZN and the Cape, were the differences based also possibly the climate, and where was the collection done? Was it along the coast or perhaps in other parts where it's deeper, not too close to the coast, because of the differences in the coastlines, the nutrients, the weather, and all of that stuff?
Okay.
Thank you.
So what we did with MISPA was to go on a road trip at the same time of year. So we targeted a particular time frame. So we pretty much went in July and drove a lot and pretty much collected within that particular season. And we collected plants in the Western Cape during that time, in the Eastern Cape during that time, and then went all the way to KZN and also collected in the northern parts of KZN and also in the southern parts of KZN, and covered areas where we could get collection permits and where we knew the plants were actually there. So we tried by all means actually to go into conservation areas. So we didn't just like pop out on the side of the road and just grab things. And even in the Western Cape and the Eastern Cape, we collected mainly in conservation areas with conservation managers to collect. So yes, the climates are very variable, and this is, one key question that I think would be interesting to try and resolve with respect to Dodonaea, because the plants are found throughout the whole entire world. They pretty much have made home throughout the whole entire world and in a variety of different climates. So we don't know at this particular stage how those climatic conditions have actually led to localized adaptations. One of the ideas that we have is that there's some kind of epigenetic mechanism that might actually be controlling adaptations of these plants to these very variable environments. So it's likely that you've got a genetic as well as an environmental pull that is responsible for the differences in the metabolome. But in a common garden experiment, we see the same patterns. So that somewhat alludes to a genetic link that's actually very much controlling or is a stamp in relation to that metabolism.
Thanks. I see Dave has a question in the chat, and he asks, what is your opinion about the potential for purified medicines from ethnobotany, or should the focus be on plant extracts?
I think we live in a system that has medical pluralism, where there are many different pathways to be able to explore plant-based medicines. I don't think that the one is better than the other. I think there are opportunities in all the different sectors because consumers go for different products. Some people say, I only want things that are natural. Not really sure what that means. And so you've got diversity in terms of consumers, and so you should be able to have diversity in products. There are instances where you isolate a single compound and then suddenly that compound doesn't have the same activity. You actually need the entourage effect. And in that particular case, then you need to go for a whole extract or an enriched extract that has, particular metabolites with good synergistic impacts. But in other cases, the case of Taxol, vincristine, vinblastine, I can name a whole range of plant-derived single-acting isolates that are sitting in the pharmaceutical industry that are used for various treatments. But you also have these single isolated products that you can actually take. It is harder to try and understand what is functioning in a whole extract because you've got so many different things that are in the extract when you compare it to a single isolated compound. But I think there's definitely opportunity for all the different ways in which you can explore natural products. I don't think any I don't think one is better than the other.
And there was another question from Adiyemi. He said, thanks, Knox, for the talk. Any suggestions on tips that allowed for the success story with the communities leading to an herbal product?
Success stories or tips in relation to...
Yeah.
I think patience, to be honest, because it's quite a long path, and often it can be difficult to negotiate with communities, especially when you come to the benefit sharing stages. I think one of our greatest constraints is just going past, getting the bioprospecting permits. It's quite bureaucratically overloaded, and at the same time, there are different expectations from different players. So I think the biggest tip is to be patient, to be resilient, and but at the same time to keep your passion so that you can see the journey to the end of the road. We need to streamline, hopefully, I think we need to streamline some of the government-related activities that are part of the pathway to commercialization. It can take a really long time. So with the Sciletium product that we have, we started putting in applications about 3 years ago, and we only got our permit about 3 months ago.
Wow.
Yeah.
Certainly a need for patience there.
Yes.
Are there any last questions from the audience? If not, thanks so much, Knox, for sharing your very patiently taken journey with us, and congratulations again on the silver medal. So
Thank you.
Certainly well deserved. Have a great evening, and thanks everybody for joining us.
Thank you. I hope everybody has a good evening, and thank you for your time.